All right, it is a few minutes past 1:30, so we'll go ahead and get started. But I'm Katie McNeely. I'm a graduate student in the psychology department here, and I have the pleasure of introducing Dr. Cassandra Gipson for her talk today. She did her undergraduate at the University of California, San Diego before shipping off to do her graduate work at the University of Kentucky, and then completed a postdoc at the Medical University of South Carolina before returning to be a faculty member at Arizona State. And it's now been a few years since she's returned back to her graduate school roots and is now an associate professor in the Department of Pharmacology and Nutritional Sciences at the University of Kentucky. She's been incredibly successful in many avenues, but also in procuring NIH and other types of funding, with at least 4 active grants that I could find and several more pending, spanning multiple areas of neural mechanisms of substance use, all around with nicotine, cocaine, opioid use, alcohol use in preclinical models, and of course has cultivated a new area of research in the work she'll be talking about today with oral contraceptives and nicotine use. And not only has she been incredibly successful in that area, but has also shown a deep dedication to fostering trainees, even those not in her own lab like myself, for who she has shown sincere generosity and provided countless resources in preparation for starting my dissertation work in an area quite closely related to her talk today. And with that, I will pass the mic off to Dr. Gipson so she can discuss her work on the impacts of ovarian and contraceptive hormones on nicotine neurobiology and behavior.
Thank you so much, Katie. That was really nice. I'm really honored to be here to get to talk to you about something that I've been really excited to work on for the past 5 years or so, I guess, is when we started up this line of research in my lab. So, first I'm just going to talk about, there we go. Okay, tobacco use disorder generally as a clinical problem, which I'm sure everyone is familiar with, but it's a highly prevalent preventable cause of death with estimates around 1 in 5 deaths due to tobacco-related diseases. And specific to women, about 170,000 women die of complications related to smoking in the United States each year. And nicotine has generally been accepted as the primary addictive component in tobacco. And although we have treatments, a lot of them are nicotine replacement. And in terms of long-term smoking cessation, they're not that great. So historically, reports on the negative health effects of smoking really didn't include women. In fact, I went back and read the landmark United States Surgeon General's report in 1964. And even though it causally linked cigarette smoking to lung cancer, it actually states right in there, this is specifically in men, less is known about women. And that's kind of been the history of, smoking in women and what we've really been focusing on. And preclinically, the majority of research on nicotine neurobiology had been conducted in males until more recently with the mandate to consider sex as a biological variable by the NIH. So smoking in women is a problem. Almost 50% of premenopausal women who smoke use some form of hormonal contraceptive, even though it's contraindicated, especially for women over the age of 35. Women are typically more vulnerable to substance use disorders, and long-term smoking cessation has been found to be more difficult to achieve in women as compared to men. And women are less likely to quit smoking than men in clinical trials for some pharmacotherapies, although results have been mixed with varenicline. Results have shown that with the patch, this is true. In terms of hormones in smoking, because women and men have very different hormonal experiences and trajectories, increases in 17-beta estradiol or E2 and progesterone are associated generally with addiction, vulnerability, and resilience, respectively, in women or in females. And oral contraceptives actually contain a synthetic form of E2 called ethanol estradiol, which is a more potent synthetic version of E2. And it's notable that EE increases the metabolism of nicotine, although the effects of this on behavior are as of yet unknown. And there's one paper from 2015 that showed that smoking satisfaction was higher in oral contraceptive users during the low progesterone week of the pill pack and lower when progesterone levels were high, showing that progesterone really may have kind of a beneficial effect on smoking in women. So just prevalence of oral contraceptive use in women is very high in the United States. 72% of women who practice contraception currently use non-permanent methods, including the pill. And so you can see in this table here that the percent of women aged 15 to 44 using the pill was around 15 to 16%, which is about 25% of contraceptive users. And in 2010, the Affordable Care Act actually mandated no consumer cost sharing for prescription contraceptives. And this led to an increase in the consistent use of the contraceptive pill by women. So why is all of this important? Smoking and oral contraceptive use can actually act synergistically as risk factors in the onset and progression of diseases such as cancer and ischemic stroke. And smoking actually leads to the earlier onset of menopause by more than a year. And in women where this has been shown to be true, they also seem to have higher mortality rates at younger ages. And smoking cessation treatments tailored specifically for women are lacking. And also in clinical trials for smoking cessation, they typically require women to be on some sort of form of birth control, but it isn't specifically hormonal or abstinence, for example. It can be anything. So the goal of our research is to examine the contribution of ovarian and contraceptive hormones on underlying reward circuitry in the brain in a rodent model of nicotine use in females. And how could this potentially translate in a meaningful way? Well, the more we understand about how hormones and nicotine interact with brain circuitry can help guide women's care and possibly treatment strategies specifically for women. So can we model this? I'm not sure how familiar you all are with how we model these things in rats, but we can actually do nicotine self-administration in rodents. And I also do something called demand using behavioral economics. We can also model the reproductive cycle in women in rats. It's not perfectly one-to-one. Obviously, rats don't menstruate, for example, but we can model the cyclical variations in hormones and changes in cycle that also happen in women. We can actually use something called ovarectomy, which clears the hormonal milieu. And it's a model of oophorectomy in women. And we can actually systematically do hormone replacement and see how each hormone kind of contributes to different behaviors, for example. And we can also look at neurobiological outcomes. So this is my typical slide I show. So basically, how do we model self-administration in rats is we do an intravenous jugular catheter surgery, and then we can train the rat to press a lever in something called an operant box, where when they press the lever, they'll get an infusion of nicotine paired with a light and a tone compound stimulus. And over time, they'll acquire nicotine self-administration. And I'll show you a little bit of data with some other things that we can do. So after self-administration, we can put them through something called extinction, where now lever pressing leads to nothing. And what you'll see is kind of a burst of responding followed by a drop off. And we can actually then give them those cues back, that light and tone, contingent upon a lever press, and see what they do. Okay, so modeling the human female reproductive cycle with the estrous cycle in rodents. Well, actually, it's thought that women have 2 cycle phases, but actually there are 4. Follicular, ovulatory, luteal, and menstrual. So obviously not all of those correspond with the rodent reproductive cycle, but there are also 4 cycle phases in rats and mice. And what you see are cyclical variations in hormones, including luteinizing hormone, follicle stimulating hormone, progesterone, and estradiol across the different cycle phases, which includes medestrus, diestrus, proestrus, and estrus. And each of these cycle phases are characterized by changes in cellular composition, and we can actually measure that using vaginal cytology. And so you can see here that as the cycle phases start to change, as the corpora lutea shrink, getting ready for egg growth and release of a mature egg during ovulation, you can actually see hormones starting to ramp up from diestrus to proestrus in anticipation of ovulation. And then those hormones will drop down. And if the released egg is not fertilized, all of this will reset back to start again. Okay, and this is just a book chapter figure from a colleague, collaborator of mine, Heather Belmonte Nelson, who you'll see her picture a lot throughout this. So how do we manipulate hormones, and modeling menopause specifically is what Heather does in rodents. We can do this with ovarectomy, which is, again, removal of the ovaries. Heather uses something called 4-vinylcyclohexene diapoxide, which actually, if you give injections of this VCD, it will deplete the follicular reserve and actually lead to more of a transitional kind of menopause as what happens in women. And you'll see, as compared to ovary intact females, ovarectomy, for example, induces weight gain. And if you give E2 back, they'll actually lose some of that weight. So weight and hormones are intricately linked. So it's a really good thing to know if you're doing any of this work, because you can actually do a manipulation check by weight and uterine horn weight as well. And so here's vaginal cytology from each of the different cycle phases, like I showed in the previous slide. And if you ovarectomize, you actually kind of wipe that cellular composition kind of clean. But I'll show you later that you actually, of the cells that are there, you'll change their composition due to ovarectomy. So I'll get back to that. Okay, so when we first started out, I hadn't yet seen a paper from Laura Odell's lab. And I was like, why won't these ovarectomized females acquire nicotine self-administration? And I was like, oh man, this is crazy. So what we found in replication actually of something Laura Odell did was that ovarectomized females were self-administering less nicotine than their ovary intact counterparts. And so we really wanted to look into this further. And so we know, based on these data and Laura's data, that ovarian hormones are critical for nicotine self-administration, but estrogens are not all the same. And actually, we don't just have E2. We also have its precursor and metabolites, E1 and E3. And we also have all those other hormones I mentioned, and they're not all the same. So they have different affinities for estrogen receptors. And you can see in this table, which I won't really get into, but there's the reference if you're interested in it, that these have different affinities for the estrogen receptors. Here are their chemical structures, and E2 is metabolized into estrone and estriol. And these are weaker estrogens that are present, and there's always kind of a ratio between these things, whereas EE is not metabolized into these weaker estrogens. So there are some pretty big differences between the synthetic and the endogenous. So we asked, do different estrogens impact nicotine intake differently? And what about the neurobiology? So that kind of led into this other study we decided to do, where we asked the question, can endogenous and or contraceptive estrogen supplementation restore nicotine intake? And so here we did ovarectomy. We waited 3 weeks to allow for acyclicity, basically giving those hormones time to kind of flush out. Then we did jugular vein catheter surgery. And on that day, these animals started hormone treatment. So all throughout recovery from surgery, they were getting hormones. And then on day 1 of nicotine self-administration, they had already had a week of their hormone treatment groups. Then they went through food training and nicotine self-administration, followed by something called the threshold procedure, which I'll get back to in a second, extinction, as I described before, and then reinstatement. And then we harvested their brain tissue for Western blot. Okay, so can endogenous and or contraceptive estrogen restore nicotine intake? Well, partially. So, all these different ovarectomy groups, even though they had already had 7 days of hormone treatment as of day 1 or session 1, they all look pretty similar in terms of consumption of nicotine as compared to the sham vehicle animals. So if you're looking at all of those lines, sorry about the ambulance, all of those lines compared to the sham vehicle group, you can see they're all much lower. And then right around session 7, 8, we start to see them dissociate from the ovarectomy vehicle group. And so we concluded that it kind of partially does. And then after that, we looked at behavioral economics. Before that, vaginal cytology was just to confirm that our hormone manipulations worked. So in our sham vehicle group, this female happened to be in diestrus, which is characterized by the presence of leukocytes. In our ovarectomized vehicle, you can see way fewer cells there, and it's diestrus-like. And then in our E2 and EE groups, you can see estrus-like vaginal cytology there with cornified cells. And so I just wanted to kind of juxtapose the different hormone groups with their different cycle phase, kind of where they would stand on that chart. So, okay, what about the contributions of other estrogens before I get to the demand stuff? Sorry. So like I showed you, E2 alone did not restore nicotine consumption to ovary intact or sham levels. So why is that? Well, so the precursor / metabolite, because you can see up in the corner, you can see how they're kind of how they go back and forth between, is estrone, which is E1. This is about 10-fold less potent than E2, but it's still more potent than E3, or estriol. It's associated actually with memory impairments, which is something that Heather found. And so we were wondering if a combination of E2 plus E1 would be key to restoring nicotine use following ovarectomy. And that's kind of what we're finding. So this is a paper being resubmitted to eNeuro by Erin Mayer, my postdoc. What we found was the presence of E1 seems critical in restoring nicotine consumption to sham or ovary intact levels. So you can see here the vehicle group, or the group in blue, compared to sham, much less nicotine. E2 isn't quite restoring it above that level yet. These animals are still running. And then E1 plus E2 doesn't look any different from sham vehicle. So we were just like, this is amazing. Okay, so how do hormones impact nicotine demand? So we can actually model this using something called behavioral economics, where we actually vary the unit price of nicotine and look at consumption. So unit price here is the response requirement divided by the reinforcer magnitude. So if our training dose of nicotine is 0.06 milligram per kilogram per infusion with an infusion length of 6 seconds, we can set the unit price of that to 1. And then if we half the dose and the infusion length is manipulated by halving the length of that, the unit price of that becomes 2, and so on and so forth. And we have 9 different doses. So now we have different unit prices, and we can look at consumption of nicotine across different unit prices. So then we actually fit this equation to the data, and we look at different parameters, including Q0, which is maximum consumption at 0 price, and alpha, which is the rate of change of consumption with increases in unit price. So just to kind of make this clear, if we see higher alpha values, it's a steeper curve, higher elasticity, or lower commodity value. And if we have lower Q0, it's lower intensity of demand. So if EE and E2 promote vulnerability to nicotine, these estrogens should decrease alpha and or increase Q0. So it's basically just transforming a dose response curve into a demand curve. So these are actually data that a previous postdoc in my lab, Greg Powell, published a few years ago. It's literally just taking, the typical dose response you see here and transforming it to these really nice demand curves. And we did this with both reducing doses between sessions. So every day they'd get a lower dose or a different dose. And then within a session, we actually wanted to manipulate this so that we could potentially do things like intracranial, manipulations or something like that. So we have a within and a between session threshold procedure that we validated. And so what we found was that ovarectomy decreased nicotine demand intensity, so that Q0 value, but it did not change essential value of nicotine. So alpha did not change. Sorry. So you can see here that where those lines would cross the y-axis, that is an estimation, which is Q0. And you can see a much lower a value of where the black line would cross as compared to the pink line. And then the 2 estrogen groups are somewhere in the middle. So we found no change in alpha, like I said, which means there were no differences in commodity value due to hormone treatment. Ovarectomy decreased Q0, or decreased the intensity of demand, and E2 and EE partially increased Q0. So a partial recovery of this measure due to estrogen supplementation. So we concluded that in an ovarectomized system, estrogens increase Q0 compared to vehicle, indicating a partial rebound. So this was exciting because these results showed specificity. And what I mean by that is that only one parameter changed as a function of hormone manipulation, much easier to interpret. So it means that ovarian hormones specifically alter nicotine demand intensity. So the next thing we looked at was extinction, like I mentioned before, where now responding leads to nothing or no program consequences. And we actually saw no difference between groups in extinction curves, but there was resistance to extinction after this threshold procedure. And we've seen this before. So Greg's other paper, don't worry about what those groups mean, but you can see here that across sessions after the threshold procedure, no group showed that reduction in responding that you would anticipate would happen. And so what we think is going on is some sort of resistance to extinction with the changes in unit price. Okay, so then we looked at Cue-Induced Reinstatement because this is a very frequently used model in the field. And what we found was that ovarectomy reduced Cue-induced nicotine reinstatement as compared to sham vehicle. And this is interestingly reversed by E2, but not EE. And we haven't really delved further into what this dissociation means in terms of responding for the cues paired with nicotine, but it seems to only be reversed by E2. And so the next thing we wanted to do was get into the brain. So how do natural and contraceptive estrogens interact with the mesolimbic reward pathway? So this is a super simplified sagittal view of some of the circuits and nuclei that we look at. So specifically, I'm going to be focusing on the ventral tegmental area and its dopaminergic projections into the nucleus accumbens, as well as the nucleus accumbens itself, because that's where we measure glutamate plasticity. But before I get into that, there is kind of a historical precedent that hormones actually impact dopamine. So Jill Becker in the 90s published this paper looking at extracellular striatal dopamine concentrations across different estrous cycle phases. And what she found was, as compared to proestrus and estrus, females in diestrus showed much lower striatal dopamine, and it wasn't too dissimilar to ovarectomized females. And I don't have the graph here, but if you give E2 back to those ovarectomized females, it increased dopamine within 4 hours. And what's even more interesting is that it was sex specific. So when they castrated males, they did not see this hormone dependent difference in dopamine. So that is even more reason that I said we should really be looking at this. So zooming into the nucleus accumbens first, I've done a lot of work in that brain area. When I was a postdoc, I kind of characterized nicotine related changes in glutamate plasticity. So the nucleus accumbens core specifically, so there's 2 different subregions that we generally look at, the core and the shell. I'm going to focus on the core. So this is about 95% GABAergic medium spiny neurons, and this is what they look like. I filled the cell with lipophilic dye and imaged it with a confocal. They receive glutamatergic innervation from a lot of different brain areas, but we mainly focus on the prelimbic subregion of the prefrontal cortex. MSNs undergo rapid changes in plasticity during reinstatement, including nicotine, which I'll show you a little bit of data from that from eons ago now. And they express D1, D2, or D1, D2 heteromers. And this is from a paper that we're about to resubmit, where it's kind of showing the output structures from these medium spiny neurons in the nucleus accumbens. So they're really important for where they project out to. And they project GABAergically to output structures. So you can see these structures include the ventral pallidum and the ventral mesencephalon, and then eventually to the thalamus, and then on out to impacting drug use. And just to kind of zoom in on the accumbens, that tripartite synapse. So in the panel A here, you can see a drug naive system where you've got these glutamatergic afferents providing tone onto these medium spiny neurons, which are postsynaptic. Ignore the astrocyte for now. I'm not really talking about that here. And then during drug-seeking behavior or nicotine reinstatement or cue-induced nicotine reinstatement, I actually showed that you have enhanced glutamate release during a reinstatement, and also that this induces rapid changes in plasticity at the postsynaptic site. So these medium spiny neurons have spines that grow very rapidly and then collapse when the responding stops. So that you can measure that both structurally and functionally. And so functionally, we measure this with electrophysiology. We measure the AMPA to NMDA ratio. This is the ratio of AMPA receptor to NMDA receptor mediated excitatory postsynaptic currents. And it's a surrogate measure of changes in synaptic strength. And right now, my postdoc Shailesh is working on some of this for us right now. So many moons ago, I showed that when you give animals just 15 minutes of cue-induced nicotine reinstatement, you can very rapidly change the AMPA to NMDA ratio. So if you look at this graph, there are yolk saline animals in white. The red bar represents animals that went through nicotine self-administration and extinction, no reinstatement. And you can see that these cells are resting in a potentiated state. And again, these are the medium spiny neurons. When you give just 15 minutes of cue-induced reinstatement, you see even higher AMPA to NMDA ratios. So meaning that just that short amount of time made these cells show rapid transient plasticity in the nucleus accumbens. But so far, this has only been shown in males. So no research today has looked at this in females, let alone the estrous cycle. And studies typically focus on this protracted time point from self-administration. So 2 weeks of extinction, driven by responding for only cues. So as a first step, we wanted to know how ovarian hormones contribute to accumbens plasticity immediately following nicotine self-administration. So when nicotine is still on board. And so we already know that ovariectomy decreases nicotine consumption, as was in Aaron's paper. And ovariectomy actually increased the AMPA to NMDA ratio after nicotine self-administration. So if you're looking at this AMPA to NMDA ratio graph, it's the purple bar compared to the black bar. So ovariectomy vehicle versus ovary intact vehicle with nicotine self-administration. This is reversed by E2 supplementation. So you can see the dark pink bar looks more like the ovary intact vehicle as compared to the OVEX vehicle. And it seems to be nicotine specific. So when you look at an ovariectomized nicotine naive animal, they do not show this enhanced plasticity. So that's really exciting, and we're looking further into what all that means. So how do ovarian hormones alter the reward pathway is still this question that we're looking at. How do they do it specifically? So we wanted to measure estrogen receptor changes with our hormone manipulations in these reward regions. So here's the nucleus accumbens core. We measured estrogen receptor alpha and estrogen receptor beta. We did not measure the GPR1. We saw no difference in ER alpha expression across the different treatment groups, but ER beta, we saw a significant downregulation in the ovariectomy vehicle group. So the pink compared to the black bar in panel C. And with E2, we saw this was reversed. EE, it wasn't quite there to significance, but it also showed a trend towards being increased. In the VTA, this is really interesting because it shows some region specificity. ER alpha, again, not changing, but ER beta was down in the OVEX vehicle group and not restored by E2 or EE supplementation. So we're looking further into the functional meaning of that. So again, what do these protein changes mean? Well, downregulation of ER beta in both the accumbens and the VTA following OVEX indicates that this receptor may be critical in mediating nicotine self-administration. But where do we go next? Well, now we should probably look at what these changes functionally mean. And so in collaboration with Dr. Brandon Henderson at Marshall University, we've been patching dopamine cells in females. So we actually bath applied the antagonist of ER beta, which is PHTPP. And we found, sorry, let me just move this real quick, that PHTPP decreased the frequency and the amplitude of spontaneous of SEPSCs. And so the next step is to determine if ER beta antagonism alters dopamine neurophysiology following nicotine, and if this is estrous cycle dependent. And so we, in addition to EPHIS, we've actually been looking a little bit more at transcriptional activity in collaboration with my new colleague, Terry Hines here at UK. So we are determining how nicotine is regulating ER beta transcriptional activity to alter neuronal function. So here we're uncovering molecular mechanisms by which E2 regulates accumbens core and VTA neuronal function when nicotine is consumed. And we have an example here using these Ishikawa human uterine cells that you can culture. So an example is TFF1 and PGR, which are ER regulated genes in these uterine cells that express ER alpha and ER beta. And we are specifically looking for genes that are expressed in the brain that are regulated by E2 and ERs. And there's more to come on this in the future, but the uterine cell line is a really nice place to start because it's easy to quickly measure these things. And so what we actually found was that TFF1 and PGR seem to be regulated by E2 and suppressed by nicotine. So if you look at these two graphs, you can see that you have your vehicle and nicotine treated cells in black and gray. And then when you put E2 in, transcription of this gene increases significantly, and then nicotine suppresses it. And to a less extent, this happened in PGR, but definitely with TFF1. So it's pretty exciting. So our future directions are uncovering neurobiological mechanisms underlying hormone nicotine interactions. Our goals are to provide insight into conditions that impact reproductive cycles and to support future mechanistic evaluations of nicotine use specifically in women, and also potential efforts to design targeted medications to support better smoking outcomes for women. So for example, in collaboration with Terry, targeting genes that are regulated by estrogen and suppressed by nicotine. So to summarize partially midway, developing treatment specifically for women is important because women are less likely to quit, like I said, smoking than men in clinical trials for pharmacotherapies we already have. And progesterone has been used as a treatment clinically for women. There are very few studies that have incorporated men, and it seems to only have efficacy for women in those studies that did, or the studies were underpowered to detect any effects in men. And compounds that restore glutamate homeostasis, which I didn't talk a whole lot about, but there's a lot of other changes besides plasticity in the glutamate system that don't work in females as they do in males. So for example, N-acetylcysteine has been heavily studied. It's had a lot of positive results preclinically to reduce cocaine, heroin, nicotine. But we found it doesn't reduce nicotine seeking in females, regardless of estrous cycle phase, as it does in males. And Lori Nacksted's lab found that ceftriaxone, which is a beta-lactam antibiotic, it restores some changes in the glutamate system. It only works to reduce cocaine seeking when females are not in estrus. So this is again when E2 is high but starts to decrease while progesterone remains low. And so there are very specific hormone-driven differences here that are important. And so, okay, I'm going to switch gears, and I do have time to show this to you, so I'm excited. So this is a new collaboration with actually an old friend of mine, Dr. Jess Weeper. We went to graduate school here at UK together. She works with women looking at hormonal contributions to mainly alcohol outcomes, but we actually got a grant together to do a translational study, so me and rats and her in humans. And we wanted to look at the impact of contraceptive hormones on nicotine and ethanol co-use. And this is a project led by Dr. Mayer. So oral contraceptives, I've really only talked about EE up until this point, but they, by necessity, contain a progestin because unopposed estrogen is highly associated with endometrial hyperplasia risk. So one of these, so there's the progestogen bigger class, which includes progesterone and progestins. And within the progestin subclass of progestogens, levonorgestrel is contained in there, and it's in a less among other oral contraceptives that are frequently used. It's also in Plan B. And so it's not known if chronic use of these synthetic hormones impacts nicotine and ethanol consumption in a co-use model. And so study one is preclinical. Our aim was to determine the influence of EE independent, or not independently, but interactively with LEVO in ovary intact female rats co-using ethanol. And our hypothesis here was that female rats given the combination of EE plus LEVO will show increased nicotine consumption as compared to vehicle or LEVO alone, which may vary as a function of ethanol experience. So this is our timeline. These animals ran for a long time. They acquired ethanol in first just one bottle, 10%, no sucrose or anything like that. And it was a drinking in the dark paradigm. So basically lights off, it's a reverse light cycle, lights off at 6 a.m. and then they got their bottles, I think at 9, and then they drank for 4 hours a day for 15 days. And then we looked at 2-bottle preference, and we kept them in the preference test until, because it was kind of an upward trajectory, which I'll show you, until it kind of seemed to stabilize. Then they went through jugular vein catheter surgery and recovery, where they started their hormone treatments. And then they got 2 sessions a day, either drinking or jerking in the dark in the morning and then nicotine self-administration in the afternoon. And then to confirm that they still preferred ethanol after nicotine, we did another 2-bottle preference phase. And so here you can see that rats pretty stably consumed ethanol. So this is the first 15 sessions of drinking here. And we got pretty decent preference for ethanol. I was really excited every day the data were coming in because it kept getting better. And then you can see pre-nicotine versus post-nicotine and the black compared to the purple lines. And after nicotine, it went up even more. So that was pretty exciting. And this is in collaboration with Josh Beckman here at UK. And this is our little secret fun study. So you know how we had rats that drank ethanol or water. I didn't mention that there was a water control group. I was curious if after nicotine self-administration, what they would do with ethanol. And so actually these animals had never had ethanol, but they were drinking water out of the same tubes as ethanol had been, the same kind of tubes. And after nicotine self-administration stopped, we saw a very high preference for ethanol. And so it made us wonder if these two drugs are serving as economic substitutes. So we're going to look into that a little further as well. So what we found with the hormone treatments is that the EE+LEVO combination group, this group showed a higher nicotine consumption in ethanol naive rats as compared to the LEVO only or the vehicle group. And then in the ethanol experienced rats, we saw the same kind of pattern. We really didn't see anything that was ethanol, ethanol didn't drive this any differently. And we're still looking at ethanol preference. I didn't include it for this talk because it was a nicotine talk, but there were some interesting hormone differences. So our preclinical conclusions are that synthetic hormones are mediating nicotine consumption in a nicotine and ethanol co-use model. In our next study, well, I'll say that later. Sorry, we have a future study planned. But our future study are essential to understand how these synthetic hormones contained within oral contraceptives interact with co-use of ethanol and nicotine. So here's the clinical study, and I'm going to do my best to describe this, but I don't work with human subjects, so sorry if I say anything incorrectly, but the aim here was to determine the influence of synthetic hormones contained in oral contraceptive pills on nicotine consumption in smokers and drinkers. And so the hypothesis here was that nicotine consumption will be higher during the active pill phase versus the placebo pill phase. The methods were that participants were eligible if they were females between the ages of 18 to 35 who use ethanol, estradiol, and progestin combined oral contraceptive pills, binge drink at least once a week, and use nicotine either through cigarettes or e-cigarettes daily or near daily. Participants complete 2 experimental sessions based on their oral contraceptives, one during their second week of active pills when synthetic hormones are consistently being delivered, and one during their placebo pill week when pills contain no synthetic hormones. And so during experimental sessions, participants complete a number of tasks and questionnaires related to alcohol and nicotine use and provide blood samples for hormonal assays. The timeline followback is used for participants to recount their alcohol and nicotine use over the past 30 days. And Jess and Ashley have modified it to fit an entire 28-day oral contraceptive cycle. So here you can see oral contraceptive users report more nicotine use during the active pill phase when synthetic hormones are being delivered compared to the placebo pill phase. This includes a significantly higher number of maximum vape sessions per day in the active pill phase. Most participants were e-cigarette users, and a trend toward more vape sessions per day and per week during the active pill phase. And a power analysis suggested that a total sample size of 22 is needed. So they're still collecting data. It was a little difficult with COVID to recruit. So here they actually had these data, which were screening data collected from over 2,000 women over the past 2 years. And they were just interested in looking at this. They actually showed that among nicotine users, women using oral contraceptives or OCs, so the OC group here, report significantly more nicotine consumption per day than naturally cycling women. So that is really interesting because a lot of the clinical studies that you may see on hormones and smoking are not actually looking at consumption. So the conclusions here was that there was increased nicotine consumption among oral contraceptive alcohol and nicotine co-users when synthetic hormones are being delivered. And future studies should continue to address patterns how oral contraceptives impact patterns of alcohol and nicotine co-use. And so our next steps, they're still, like I said, they're still actively collecting data to determine if specific types of oral contraceptives differentially contribute to nicotine and or alcohol consumption in women. And then in my lab, in collaboration with Josh, we're using a rodent model of co-use where we're going to actually determine neurobiological consequences of contraceptive hormones on both ethanol and nicotine consumption. And this time we're going to use a multiple schedule in the operant box instead of this, in the home cage drinking in the dark procedure. So there are some other future directions for us. Since I have a little bit of time, I'm excited I get to at least briefly show you. So in collaboration with Jess Santolo here at UK, she actually developed these adeno-associated viruses that are, so basically it's an shRNA packaged in an AAV that knocks down ER beta, and she validated it in a number of brain regions. She also has viruses for alpha and GPR1. So we are putting them into the VTA, and we're looking at nicotine demand, VTA dopamine excitability, and nucleus accumbens glutamate plasticity. And Aaron took these beautiful confocal images of estrogen receptors actually located on dopamine neurons, so you can visualize that. And then Emma Bondi, in my lab, she is really interested in looking at microglial morphology. So a whole other line of research in my lab is looking at neuroinflammation as consequences of chronic nicotine use. So we're really interested in studying estrogen receptors and progesterone contributions to neuroimmune signaling in females during nicotine consumption, because microglia actually express some of these receptors, and hormones can impact neuroimmune signaling. And so we have a really cool transgenic rat line that allows us to control microglial activation that she's been validating. She's been working very hard to do this. And she actually implemented an automated analysis for microglial morphometry that takes out any kind of user bias. And so we're going to be doing that in the future. And then I have one more fun little side study that I've been working on with a friend of mine, Paul Whitaker, who is an expert in nicotinic acetylcholine receptor measurements. And so I actually took out ovaries from female rats that were different ages, and some had nicotine and some didn't. And we're characterizing basically nicotine, acetylcholine receptor, and links, which is alters nicotinic acetylcholine receptor expression. We're looking at all these different outcomes in the ovary. And what we found was that ovaries express nicotinic acetylcholine receptors, and he was really excited because there's a rare subtype that seems to be present on ovaries, the alpha-6 beta-4, which may co-assemble with alpha-3 or beta-3, and also alpha-7, which is pretty ubiquitous. But so those are ones we definitely found. And why is this interesting? Why am I the crazy lady looking at nicotinic receptors on ovaries? Well, smoking leads to early onset of menopause. And so we're really interested in the question, what if nicotine is inducing early menopause directly at the ovary through its actions on nicotinic acetylcholine receptors to facilitate follicular depletion? And so there's more to come on that. We're actively doing some stuff with that. So I just want to say thank you so much for listening to all of the stuff coming out of my lab. I've got great postdocs, Aaron and Shailesh, technicians in my lab, Emma and May, all of our undergraduates who are amazing, and all of my collaborators who put up with me, and then NIDA and UK and the Arizona Alzheimer's Consortium for funding. So thank you.